On behalf of Cambridge Innovation Institute's Global Web Symposia series and our sponsor, Eurofins DiscoverX, I'd like to welcome you to GPCR signaling bias, a valuable and accessible property of new drug candidates. My name is Elizabeth Lamb, and I'm the technical host for this event. Now I would like to introduce our presenter. Terry Kanakin, PhD, is professor of pharmacology at the University of North Carolina School of Medicine. Terry is a GPCR key opinion leader, trained in chemistry and pharmacology, and he has spent 25 years in drug discovery at GlaxoSmithKline before joining UNC Chapel Hill, where he teaches and leads research in receptor pharmacology. Welcome, Terry. The webinar is yours.
Thank you, Elizabeth. This is a webinar on receptor signaling bias, and I'm going to try to convince you that it's a valuable and accessible property of your compounds in your discovery project. So what we will be covering today, we'll first talk about what is bias, and then what causes it, and then how would you use it in your program, how to see it, how to know it's there. And for that, we will show you an easy way with a tool called a bias plot. No equations, no anything there. But then if you want to quantify it at the end, we will talk about a simple equation, a simple procedure to quantify it with a number as well. So I'd like to start by pointing out ground rules. Bias is there. It's an outcome of natural allostery. So to ignore it, you're just walking away from something you already have, and that's probably the most important thing. Knowing that you don't really have to seek it out, it's already in your program. What do I mean by that? Well, allosteric probe dependence is a common feature of all allosteric protein function. Say I have a protein here in panel A, and two tracers, molecules, interact at two sites. If I add an allosteric compound, I change the shape of that protein. There's no rule that says the change in shape should be equal for tracer 1 and 2. In fact, the experience usually is it's not. So you'll affect tracer 1 more than tracer 2. Well, if I were to take those sites, put them inside the cell, give them names like G protein site, beta arrestin site, that's all bias is, probe dependence that happens naturally for each of the signaling pathways. So again, I'll just state, should your program seek bias? I am often asked this. No need. Bias will seek your program. And I don't know of a receptor system that does not show compounds that are biased. That's not proving, of course, that all of them will, but so far things like small compounds for GPCRs, peptide agonists, there we have terzepatide, peptide antagonists, antibodies, and allosteric compounds all practice bias. This latter one is something that I'll return to at the end of this talk. It's a topic we can't expand upon, but we could at a different time, where all allosteric compounds actually induce bias in natural signaling. So just to continue with the ground rules, bias assays detect ligands that make stable, unique receptor active states. I'm often asked, well, what if I'm not interested in GS versus beta-arrestin bias? I'm interested in other pathways. That's not the point. Bias assays just detect the compounds that produce a different active state, and that can play out various ways. And so once you have your compound, next step would be to test it in your system with your natural systems to see if that plays out some way that is advantageous. And we'll talk about it can be exploited therapeutically, and it is very simple to detect and quantify. And the key there is assays. So we will talk about assays. The whole key is if you have an assay to see a signal, you can apply it to this and measure bias. And I'll specifically be talking about assays from Eurofins, DiscoverX, that have a range of assays. Very important because GPCRs are pleiotropic. They interact with numerous G proteins, beta-arrestins, and so forth. And this gives compounds that stabilize an active state a whole range of signaling capability. So just a few examples when I talk about assays, the kinds of signals that we can quantify: second messengers, cyclic AMP, IP1, calcium, beta-arrestin, or various behaviors of receptors, internalization, going to various parts of the cytosol, interacting with ramps, just about anything that the receptor can do that a compound can modify, we can measure and we can capture bias. Now, here are a few of the great assays that Eurofins has for second messenger, cyclic AMP, hit hunter, calcium, beta-arrestin recruitment, interactions with various G proteins, and then measuring where the receptor goes in the cell and what it does when it goes there. So these are the important tools that we apply to try to capture the bias of our compounds. Before all this became commonplace, efficacy was thought to be a single signal varying in strength. Now we know it has quality of some GS, GI, beta-arrestin, what have you. And so a compound presents an array of efficacies to the cell. Here we have an example of how that plays out. These are 16 opioid agonists run in 6 functional assays, and we took the data and clustered it with a program that clusters genes. It's from Harvard. It's called Morpheus. And what Morpheus did was it said, here is what the cell sees. The cell sees these agonists as similar in terms of the signaling profiles according to this pattern here. And what is interesting is the cell sees things like dynorphin A and morphine very structurally different, but sees them as pretty equal in terms of the signals that they give. So the important thing here is every agonist probably presents a unique fingerprint of efficacies to the cell. And this is what your program can capture with these tools. So what is bias and what causes it? I'll give you an example. Angiotensin normally signals and activates GQ protein and beta-arrestin, but you can have an analog in Travina 120 it'll interact with the same receptor, the same cell, but only activate beta-arrestin. So now it biases the signal, completely changing the natural pattern produced by angiotensin. And we can use this therapeutically. How does it do it? What's happening is these compounds stabilize various active states. They enter the ensemble of possible states, and they prefer some over others. So agonist 1 may cause a state that will interact with G1. Here, an agonist 2 cause a state that will interact with G2 beta-arrestin. They're different, and this will cause different patterns of signaling from agonist 1 and agonist 2. So how do we apply this, use it in our discovery program? We all know that agonists can interact with various receptor type, but then within each type, we have differences in signaling patterns. So just think of it as another layer of selectivity. So we can use this in various ways. For one thing, we can emphasize a signal that we want. For example, beta-arrestin could be an important signal from PTH for the treatment of osteoporosis. And so we might want to bias our compounds towards that signaling for that therapeutic output. Or we might want to de-emphasize a harmful signal. For example, morphine will depress respiration, but it'll do it less in a beta-arrestin knockout mouse. So this would suggest you'll have a better, safer analgesic if it's biased towards G protein and away from beta-arrestin. Or you might want to de-emphasize a harmful signal and also prevent the natural system from doing it. And that was the example of Trevena that I showed you earlier. Trevena 120 will do the good thing that angiotensin does and take away the harmful vasoconstriction in cases of heart failure. It may also allow us to go after targets that are prohibited. Here we have some great work done by Kate White. Kate showed that kappa opioids produce many effects that you could construe to be useful in cognition, reward, mood, perception. However, the trouble is, agonists for this target, like Salva, also produce horrendous hallucinations. Kate showed that that was probably due to the interaction with beta-arrestin. So if you could get a biased compound away from that, you might have a useful kappa opioid agonist that you could use therapeutically. One of the most common ways we apply bias is to perhaps we don't know what we want yet. Back in the old days, we would run a screen and we would take our most active compounds and move it on to the next assay. Now you can cross screen, take your actives and run them in a different assay. And I guarantee you, you will get a scattergram. The compounds will not be equally active in each assay. That is what bias is. However, now what we can do, we can pick out the compounds that we know are forming different active states, and those are shown in red. The chances of us getting a unique phenotype in our next assay are probably much higher with these compounds than if we'd have simply taken the most active compounds from our screen, which could be doing the same thing. So you would cross-screen and use these to try to pick up unique physiological phenotypes that could be therapeutically useful. So, how do we identify bias? It's extremely easy without equations, models, or anything. You simply say we have an agonist that signals in two assays and call them pathway 1, let's GS, cyclic AMP, pathway 2, beta-arrestin. You then express each response as a function at the same concentration and form what is called a bias plot. So all I've done here is I've taken the common concentrations and say the concentration that produced this much response in pathway 2 gives me that much response in pathway 1, and we get this curved plot. The fact that it's curved does not mean bias. So all that might mean is that the pathways are different. For example, we all know that cyclic AMP assays are more efficiently coupled than beta-arrestin assay. That is system bias. So let's say we had two agonists and we do this and we get those bias plots. All this means is those compounds are producing the same active state, and that difference is system bias. What you're looking for is the compound that jumps out at you. So now we have a different compound, and its bias curve is different from the bias curve for this agonist. That states that compound's creating a unique active state from that compound. An example. So this is the portable parameter that will identify the compounds that'll be biased in all systems. An example is shown here. These are mu opioid agonists. Bias plots for these agonists are by and large equal. Carfentanil jumps out at you. This assay tells us that this compound's active state is different from those. And so this is the easy way. This is some data from Eurofins where we looked at various chemokines, and we see that there are two groups here, CCl3L1 and CCl5 produce a different bias, different active state from CCl4 and CCl3. Well, let's say we want to put numbers on that to tell chemists how much more bias a certain compound is over a different one. A good scale to use comes to us from the operational model, which quantifies efficacy and affinity. And we just simply do that for each signal. So I can quantify this number, which is the ratio of efficacy divided by affinity for G protein 1 and then beta-arrestin, and use these numbers to compare. And I'll show you how we do that. We have to use log of efficacy divided by affinity. We can't just simply use potency because of what happens to potency in different cell types. Here we have weak, low efficacy compound run in a number of cell types, and what I've done here is I've just run them in ever increasing receptor expression levels. What you would see is the curves would go straight up, and then once you max out and everything's a full agonist, then it starts to shift. It's a different behavior in different cell types, so you have to correct for the differences. In fact, the operational model was created to handle this because before it, you could not compare the potencies of a full to a partial agonist. You couldn't use the EC50. However, once you convert and get log efficacy divided by affinity, you could. So now here's an example. This is Eurofins data, and we have exendin and various agonists for GLP-1 receptor cyclic AMP and GLP-1 receptor beta-arrestin. A surrogate for log efficacy divided by affinity is log maximum divided by the EC50. So the first step, there's 4 simple steps here. Get your maximal responses and your EC50s and calculate log max over EC50. This is also called the transduction coefficient. Think of it as a single number that quantifies the power of an agonist to produce response. Once you have that, then you rank it within each signaling pathway. You still can't cross over from pathway 1, cyclic AMP beta-arrestin, because the receptor expressions and so forth are different, so you can't compare those. What you do is you rank them within each assay. You choose a reference compound. In this case, we chose exendin. And now we have the logarithm of the relative power of these agonists within each assay. Once I do that, I can cross over and I can calculate bias, which I've done here. And that is delta of delta log max over EC50. And then take an anti-log. What this says is, in this case, these two agonists show a 17-fold bias towards cyclic AMP over beta-arrestin compared to the standard exendin. This is the number that the chemist will use to gauge the relative bias of a compound. It's selectivity, but it differs from standard receptor selectivity, which is you won't get the effect on the secondary receptor till you get to the concentrations that start to activate it. So you will have a concentration range. Bias is occurring the minute the compound interacts with the receptor, but at different strengths. So both signals occur once the compound binds, but if we have a bias towards cyclic AMP, the cyclic AMP signal will be much stronger than the beta-arrestin. So how that shows up, for example, here we have the Eurofins data, cyclic AMP, this is for tirzepatide, and beta-arrestin, and I simply put the curves on a common axis. Does this mean that the compound won't activate beta-arrestin until you're long since bound to the receptor and are producing maximal cyclic AMP responses? No. What this means is that these two assays are of differing sensitivity. So when we try to quantify receptor selectivity, for example, this is data from Hager where we have peptides interacting with GLP-1, GIP, and glucagon. The receptor selectivity of this peptide is very strongly towards GLP-1. So the concentrations that activate GLP-1 do not do anything until you get to hundreds and hundreds fold higher, and then you see this. But that same peptide, if you're comparing bias, is activating all of these pathways once the compound binds to the GLP-1 receptor, but at different strengths. In this case, it is biased towards cyclic AMP because those signals are stronger than the others. So it's easy to detect, it's easy to quantify. The real challenge then is the translation. And this is where the field is now. It's well and good to get these compounds, but we have to somehow quantify where it's real and useful and use that therapeutically. And actually, that the history of it is there are compounds that have failed in this translation state. For example, Travina 120, which was good in vitro but did not have the properties required therapeutically in vivo. Why does that happen? Because think of it as this is the example, the analogy I've chosen here is a cipher machine that was used in World War II called Enigma. And so you would key in your message here, and then each letter was randomly scrambled to a plug board and then scrambled again through 5 turning wheels. And each day they would change these wheels. And so what came out was very different each day. And this was thought to be well nigh unbreakable. You have 159 quintillion possibilities. That's really what could be happening when you put in a biased compound and it's interacting with a range of cells with a range of different stoichiometries between couplers, G protein, beta arrestins, and so forth. That scrambling can produce these kinds of differences. So what you measured in vitro in your system may translate to be something else different in vivo. I'll show you an example from our work years ago. We were interested in the calcitonin receptor, which was expressed in a hex cell. And so we would have a relative potency of porcine calcitonin to amylin. And then we did the same experiment in a hex cell. And the only thing we did was we enriched that hex cell with added GS protein. Now, these compounds are differently biased towards cyclic AMP over other signals. Well, when we enriched GS, we selectively increased the activity of porcine cal over amylin. So just changing the cell type changed our bias. And so changed it by 46-fold. This is the challenge we now have. We have a biased compound, but we now may see differences in that bias as we go in vivo therapeutically, which is one thing to argue for taking it into the most therapeutically relevant natural assay you have as quickly as you can. Finally, as I mentioned earlier, all allosteric compounds that modify signals can and probably do induce bias. And so you will actually change the quality of efficacy of your natural signaling just by putting in an allosteric compound. Would be a good topic for a future webinar. So I'll stop there and I'll summarize. Bias is not optional, it's happening in your program anyway, and it can be exploited for therapeutic means to enhance or depress certain signals. The key here is to have the assays to detect it, and that's easily done. You can also quantify it with simple models. And so I'll stop there. I'll direct you to, we have a white paper on this which outlines all of these concepts with the examples I've shown, and you can access that at that URL. So at this point, I guess I'll stop and see if anyone has any questions.
All right, thank you very much, Terry. We will now open up the question and answer portion of the meeting. Moderating our Q&A today is Dana Haley Vicente of DiscoverX.
All right, thank you, Elizabeth and Terry, for your very nice presentation. The first question today is, do we talk about TAR receptor, so TAAR1?
Well, actually, I have had no experience with TAR1, although I have heard of it, but it shouldn't matter. I mean, all these concepts are generic for any target, so all you really need is tools, compounds that interact with the target, and then you can compare potencies. It'd help if we had more texture on that question to see what exactly our question means.
Okay, well, I'll continue with another question. Maybe we can get more.
Okay, alrighty.
The next question is, would it be preferred to run primary high throughput screening with parallel pathway screening, or only profile bias after hit identification? I can read it again.
Oh, good question. First of all, I don't know that you would run cross screening on your whole library. Usually, what's done is you run a screen and you get a subset of compounds that interact with your target, and on those you would then do a cross screen. The advantage of that is you will identify compounds that are biased. If you go ahead and choose a lead, you may have chosen one that does not have the bias that other compounds do have, and you didn't see. So I would run the cross screening on a subset of compounds and use that to choose compounds that are biased, and then move those forward.
Are you ready for the next question?
I am indeed.
Okay, this one's quite long. Okay, it says, there are now many biosensor platforms available to investigate biased GPCR signaling. However, results obtained with different biosensors can sometimes differ considerably, even when the same agonist, receptor, and cell line are used. So the question is, in your opinion, how should such discrepancies be interpreted?
The real world. Thank you. Yeah. Well, as I tried to point out the choice of your assays and signaling pathway is almost immaterial. These are just tools to reveal which compounds stabilize a unique active state. So as that happens, whatever pathways you've got there will read those differently and point out that compound. You were right. What you're actually talking about is translation. Systems can then change that. And there's a rich history of what is the best way to do it. Measure bias, numbers don't agree. Is it an artifact? I think it's an imperfect world out there, and we're to the point where we just use these tools to point us towards probes, unique compounds of interest, and then get them into the most physiological systems we can, the systems that we have an interest in. What brings to mind is there is a wonderful new system out there called TruePath, where you can measure the interaction of the receptor to 21 or 22 G proteins all at once, and you see varying patterns. They're all different. So this is just a qualitative answer to say this compound is producing a different active state from another compound. Let's go see what it'll do in the relevant system thereafter.
Okay. So the next question is, the standard assays to detect bias traditionally are G proteins and beta-arrestin. What if I'm not interested in those pathways for assessing the bias of my molecule?
Right, and that's sort of follows from what I just said. Again, it's just a tool to point you towards molecules that stabilize an active state that is different. The question you are most often asking is, does my synthetic agonist produce a signaling fingerprint different from the natural one? And if so, how different, and are those differences good or bad? So the most common ones are G proteins, beta-arrestin, but now there are all kinds of assays, and there's all kinds of bias as well, signaling bias, location bias, and so forth. So it's just the question of pointing us towards unique agonists that we can study further.
Okay. It says, do I need to assess bias in the same cell?
Ah, not at all. No. I'm often asked this. You're ranking compounds within each assay. So you have a reference and then test compounds. So in that sense, everything's being run in the same cell. Once you've done that, then you can cross over cells and you can use any assay. Obviously, a cyclic AMP assay is a lot different from a beta-arrestin assay, and the cell type is really immaterial.
How much bias would be therapeutically relevant?
Oh boy. good question there, because that's going to probably be system dependent. For example, I'll give you an example that we did at GSK. We were interested in a GLP PAM for diabetes. So it's the same kind of question. How much potentiation of GLP-1 do we need? Same question as how much bias do we need? In that example, we had an external standard that really helped us. There is an enzyme that destroys GLP-1 called DPP4. And DPP4 inhibitors produce about a 3 to 4-fold elevation in GLP-1, and that was enough to cause a useful effect. So there we had an easy one. If we can get a 4-fold increase, that probably is enough. But you don't often have that. You have no idea. I'll give you the example of M4 PAMs for cognition. If you potentiate M4 response, you might improve cognition in schizophrenia. And so people went with PAMs that gave you 200, 400-fold potentiation, whopping hammers. And of course, they got cholinergic side effects. So you don't know how high. Valium is a powerful drug. It produces about a 2.5-fold potentiation of the system, but that's enough. Calcium. Cells hate calcium. You give a cell too much calcium, you'll kill it. So the calcium sensor receptor PAMs for that, producing a 10-fold, 12-fold potentiation, those compounds are toxic. The approved drug, cinacalcet, produces a 2.5-fold effect. That is enough. So the short answer to all that is it will be system dependent. I'm not sure. I think biology operates on the steep part of the curve, and I don't think you need these huge effects like 10-fold, 100-fold effect to change it. So the hope is a nice mild effect will be enough.
Okay. Do we have another question that says, many thanks for your inspiring talk. What is your opinion on using barcoded assay systems to measure GPCR-based signaling?
Now you got me. I'm not entirely sure, Michael, what a barcoded assay system is. So I would love to answer your question, but I'm at a loss here. If you could expand on that and we could maybe return to it, that'll work.
Okay. All right. Another question is, thank you for your explanation. What do you think about bias antagonists? Recently, I've read several papers describing bias antagonists and find the concept very interesting. If you may ask again, or if I may ask again, do you also mind to provide must-read literature for or about bias?
No. Good question. Okay. First of all, you have to define what you mean by an antagonist. An orthosteric one, I cannot see how you could ever have that happen, because an orthosteric antagonist simply precludes agonist binding. So whatever the agonist does, whatever bias it has, you'll stop it and you won't change it. However, allosteric NAMs are famous for producing bias. Anything that changes the shape of the protein technically could change the quality. So you have compounds, and I'm struggling trying to remember some examples. There's one where the allosteric antagonist only inhibits beta arrestin coupling to the receptors. So you transform natural G protein beta arrestin signaling to only G protein signaling. This is common, and with NAMs, I think people are going to have to check this, because you can't just assume you are inhibiting the response equally. There is a good chance you will induce a bias. And again, this must read. Well, it's a hot area. What I would, there are a few articles out there. One you might read, and it's the pharmacology review I wrote, and I'm not citing it because it's really good or anything, but I did take time to survey about 600 articles and methods and so forth. So it gives you a spectrum of what's out there. Having said that, it's getting old. It's about 5, 6, 7 years old, and this field is moving very rapidly. So I would just keep an eye out for the most rapid ones. If anyone is interested with for specific articles, email me separately, and I'd be happy to tell you a list.
That's a good idea. Okay, we have a time for a few more questions. The next one is, it's a follow-up of the
barcode. Thank you, Michael. Okay. Okay.
You want me to read it?
Okay. Yeah, please. Yeah.
Okay, it says barcoded assays use short mRNA barcode reporter instead of a standard reporters like GFP or luciferase. Recent examples are Presto, Salsa, using beta arrestin full TEV technology, or the GPCR profiler, using the beta arrestin split TEV technique.
It's a changing world, man. That's great. I am not sure about the details differentiating it. Technically, anything that tells you a difference between the agonist bound receptor with the signaling system is useful. It's just telling you you've changed something. So I would, some of these assays though, like TruePath, possibly label free and stuff, you're getting answers that are so textured that they're also getting difficult to interpret. And I'm wondering if this might start to get into that realm as well, where you get such a textured answer that every agonist will be different. Having said that, I think you could support that pretty well every agonist is presenting a cell with a unique fingerprint of efficacy, and it's a question of how different, and if a certain difference will change physiology. But this whole idea of bias, think of it, it's based on a standard physiological law. called allosteric probe dependence. It's happening all the time. It's been happening for hundreds and hundreds of years. We just didn't see it till we had the assays to see it. And so it's new to us, but of course it isn't new. So it's the assays that are the important thing here, that discern that this compound is producing a different quantification of physiology than another compound.
Okay. We'll wrap this up with one more question. Bias is selectivity. How does it differ from receptor selectivity?
Right. This is a sometimes confusing thing. If you think of receptor selectivity, let's say we have a compound that has an EC50 for receptor A of nanomolar and an EC50 for receptor B of micromolar. There will be 1,000-fold difference in the concentrations that will start to induce signal B compared to signal A. Yet you'll have that separation. Bias occurs for everything at once. As soon as the compound binds to the receptor, if you're asking a question, what's the difference between G protein, GS, GI, GQ? All of those kick in. Once the compound's on board and you've changed the state of the protein, it will interact with GS, GI, GQ, but with a very different strength of signal. So you might start GI, but it'll be so weak, it'll be in essence zero, and you need to give more compound until that finally kicks in. But bias will happen simultaneously at varying strengths, and the effects I predict will be more subtle. I can't see biases being hugely, like hundreds and hundreds of fold, which you can see with receptors differences, but we will see. Having said that, beware of perfect bias. There are compounds you can say, well, there are compounds that will induce one signal and not induce the other one at all, like Trevena 120. GQ, it doesn't turn on at all. Beta-arrestin does. So it has been called a perfect bias. That's only true because your assay wasn't good enough to see what was happening. Something's happening there, but it's probably so weak you need a cranked up assay to see it. So you cannot quantify bias unless you have two curves. If you don't get a curve for one of the signals, you're pretty well out of luck. You can't really assess bias because in a more sensitive system, you might get surprise. That so-called perfect signal will appear.
All right. Well, thank you, Terry. If there's any more questions that did not get addressed, it'll be answered by Terry after the event. He'll receive these questions, and you'll also receive an email with a link to the webinar shortly. And thank you very much. We're going to go ahead and close the webinar.
Thank you, guys.
Bye.
Bye.